everybody. Thanks for coming. And it's a great pleasure to introduce Natalie, who's the reason why we're doing this talk is because Natalie, Toby and I were in Adelaide, and Natalie gave a talk there, and Toby said, I never actually understood what you did. It was fantastic to hear about it. So I thought it would be good for people who are in the lab, and also great to see Barrett and Jared here. Thank you for coming along. To find out a little bit more about Natalie's research and her ideas and her projects. So Natalie's had a Synapse Fellowship here in SensiLab, and she's doing a PhD at RMIT that I'm supervising with Roland from RMIT in architecture. But the work that she does is very interdisciplinary, and it crosses over from biology, robotics, digital fabrication, and architecture itself. So thank you, Nat, for agreeing to do the talk, and I'm sure everyone will have a completely new perspective on what you do after hearing the talk, so thanks.
Thanks, John. I actually want to do a show of hands so people actually know what I do. But anyway, thanks, John. So today's presentation, I'm just going to give a quick run through about my background, basically what my PhD is based on, and current research that I'm conducting. It would be great to hear your thoughts, questions at the end. So I'll just present my PhD research topic, which involves the symbiosis of machine and organism. And this basically is a focus on the fusion between the three topics of computational design, biological materials, and robotic fabrication. And what I'm quite interested in is how these three mediums, which are ordinarily treated as separate disciplines, actually interact with one another to create this new multidisciplinary approach to architecture. So before I get into my research, I'll just give a quick timeline about my background. So I actually completed my master's in architecture in Barcelona in Spain at IAC, and IAC really specializes in robotic fabrication and parametric design, and that's where I really became interested in this multidisciplinary approach to architecture. I'm not sure if anyone's familiar with Marcos Cruz, but he's basically the pioneer or godfather of fusing science and architecture together, and he supervised my master's project, which is based on mycelium, and that's how I really became interested in this topic. So when I returned back to Australia, I worked as a graduate architect for Ellenberg Fraser. But this passion for biology and architecture always remained within me. So I returned back to Monash and began to run BioLab Studio. This is a design studio where students focus on scientific materials and how to apply that to larger architectural scales. And then I was introduced to John and started research assisting here at the end of last year. Yeah. And then from that research, we applied together for the research grant that's called Biohybrids. And then began my PhD with Roland Snooks, and I'm now conducting BioLab here at RMIT, here at Monash and RMIT. So basically, as part of any PhD, I'm currently conducting a community of practice, which basically really focuses on where the contribution of knowledge within this broad field of architecture and designers who infuse architecture and science together. So I'll be looking at topics such as biomimicry, architects utilizing green facades, but where I think my main contribution to this existing field is architects utilizing biomaterials at a one-to-one scale, and architects really allowing the chemical reactions of organisms to really drive the design process. So I'm currently focusing on a range of architectural tectonics that have evolved from these scientific experiments. So what I'm quite interested in is, rather than creating static architecture that's ordinarily derived from materials such as glass and concrete, how do we create architecture that's similar to biology? So something that self-repairs, responds to the existing environment, ecosystem, and grows and adapts. I think it's cut off, but anyway. And therefore, this brings me to my focus of these three mediums of biological materials, computational design, and robotic fabrication, in order to focus on this research question of creating adaptive environments through architecture. So back to this Is there a way to fix this or no?
What's happening?
Oh, it's just been cut off. It doesn't matter. It's no big deal.
It's been cropped. Okay.
It doesn't
I
matter.
would assume that's your screen. There's no.
Okay. Anyway, it's just like a little bit anyway. So, is that me? Okay. Anyway, it's okay. So part of my community practice, I'm currently looking at existing precedences, so researching medical journals, researching architects utilizing biology, researching many designers fusing robotic fabrication and natural organisms together, so actually 3D printing with a number of living materials. But what I'm quite interested in is this specific field of architects utilizing natural organisms at a one-to-one scale, so architectural scale. And through this research, what I really concluded was architects really treating the scaffolds and living organism as separate mediums, so robotically fabricating something, infusing a living material, and therefore neglecting this crucial relationship between the two mediums. So what I'm quite interested in is creating this constant dialogue between the two, so how can the organism have a direct effect on the robotic scaffold or vice versa. Which brings me to the work of Neri Oxman, who's, as John knows, is quite an inspiration of mine. So her methodology of really allowing the materials' patterns of growth and chemical characteristics to really drive the design process and therefore final design outcome of the overall prototype. However, through this research, what I found was, once again, this almost like unexplored territory of the secondary skin and static scaffolds being treated as two separate mediums. So I'm sure everyone's familiar with the Silkworm Pavilion. And it's...
Actually, no, sorry.
Yeah.
Everyone will be familiar.
Oh, sorry. Okay. Thanks, John. So,
Never heard of it.
okay. So Neri Oxman is a researcher from MIT. And basically, she's currently conducting research and many large-scale prototypes, which really focuses on biological materials and robotic fabrication. So the Silkworm Pavilion is basically, I don't have an image of the final, but a large-scale geodome. She then applied the silkworms on top of this geodome to create this secondary skin. So silkworms basically spun their silk and created a secondary layer on top of this robotic fabricated scaffold. And my critique on this project is that the silkworm skin and the scaffold are treated as two separate elements rather than having a direct influence on one another. So which brings me to my research, which is mycelium. So I'm sure most of you have seen the mushrooms around here. So mycelium, for those who are unaware of the organism, is the root of a mushroom. It has its chemical reactions include its ability to act as a natural binder between two materials, its ability to remove toxins from water, and its ability to biodegrade through complex scaffolds in order to reach its fibrous food source. So mycelium is actually not a new material within the architectural discipline. Many architects have utilized mycelium at a larger scale in terms of its ability to replace concrete. So mycelium actually works extremely well in terms of compression. So many architects and designers have utilized this at quite large scales. So my intention is to really focus on this ability for mycelium's ability to biodegrade through a number of scaffolds. I've mentioned previously that a number of architects utilizing natural organisms really treat the organism and the scaffold as two separate elements. So through this research, what I'm quite interested in is how the mycelium can actually biodegrade through a range of set geometries. So this ideology and methodology of biodegradability currently exists within the scientific medical industry. I'm currently researching these techniques such as 3D printing organs, bioprinters, bioscaffolds. And what's quite interesting about these existing methodologies is how scientists already have created this fusion between robotic fabrication, 3D printing, and biomaterials. So my intention is to really look at these existing formulas and start to create a multidisciplinary approach to architecture. So start to think about how do we apply these technologies occurring within the scientific microscale and apply them to the architectural macroscale. So for example, how bioscaffolds work within the scientific realm is that the cells are extracted from the human, the form is computationally designed, and then the cells and form are fabricated simultaneously on a bioprinter. So what I find quite the most fascinating about this process is how the scaffolds and the living cells actually interact between one another and have a life of its own post-fabrication, which is quite different to architects. Once we fabricate something, it's a finished process, we move on to the next project. However, within science, when they utilize this methodology, it's basically how the scaffold reacts to its host system. That's where the ephemeral changes occur within the prototype.
Can I ask a really quick question?
Yeah, sure.
Can you just explain what's happening there?
Yeah, sure. So this is actually the cells being adhered to the bioscaffold, and basically the scaffold is absorbed and the cells are overtaking the form of the scaffold. Yeah. That's a sheep's heart, by the way. Yeah. So as I mentioned, my intention is to really investigate these existing technologies occurring within the scientific fields and think about how to actually apply them to architectural buildings and architectural facades. So this concept of how to create something that may adapt and react to its host system, which would ordinarily be the human body, and how to take that methodology and create something that would adapt and evolve according to its architectural host system at a large scale. Which brings me to my current research. So I mentioned previously, I'm focusing on mycelium and its ability to biodegrade through complex scaffolds. However, what I believe my main contribution to the existing chemical reaction of biodegradability is my ability as an architect to really control and orchestrate this process of biodegradability. And I'll be doing that through a few mediums, which I'll showcase today. So currently I'm experimenting with a number of mycelium species, heterogeneous skins and forms, testing a variety of complex geometries that may really alter this choreographed process of biodegradability, creating multi-material, multi-scaled prototypes, and of course, really thinking about site-specific forms, so what the actual architectural application may be for the final result. So currently I'm experimenting with a number of mycelium species, just to really test which species really thrive in terms of their tensile and compressive strengths, and which species actually thrive in terms of their ability to biodegrade through scaffolds at a more rapid pace. And what's quite interesting with this process is creating one unified form, however, inserting a variety of species at a variety of anchor points, and then really documenting the rapid rate of degradation of each form within this singular skin. May I do that? Is that better? Yeah. So currently I've been experimenting with how to really improve the chemical characteristics of mycelium. So mycelium actually has proven itself to work quite well in terms of compressive strength. Currently I'm testing how to improve its tensile strength by adding a number of additive materials to the mycelium mixture. I'm also experimenting with a number of mycelium mediums. So rather than just experimenting with the mycelium paste, I'm also experimenting with mycelium gels. And this process has really been derived by extracting the cells out of the organism, mixing it with a range of agars, and then inserting this mixture back into the scaffold to test this process of biodegradability. So as mentioned, I'm currently attempting to hack almost into the chemical characteristics of mycelium. So I'm adding a number of natural organic materials in order to improve its tensile strengths as well. So currently I'm testing coffee grounds, hemp fibers, sawdust, in order to create this customized hybrid of this super material almost. I'm currently experimenting with how to robotically extrude these mixtures as well. And obviously a number of researchers and architects have been experimenting with how to robotically extrude organic materials. So I've been looking at this unexplored territory as well. Here's just an image of the mycelium extruded out of a basic syringe. Hopefully in the near future, I'll be able to take this methodology and put it to the UR robot or another robot to do this robotically rather than manually through a syringe. And here's just an image of creating a number of mycelium mediums as a tapestry to see how these living materials bond and interact between one another. And just some images showcasing how once extruded, the thing is still alive. So it actually reacts and adapts according to its host environment and existing ecosystem, which to me as an architect is quite fascinating because once I fabricate something, it's almost a finished process. But in this case, it's an ever-evolving process once the fabrication process is complete. So I'll just mention this one in one particular experiment, the BioScaffolds. So currently I'm experimenting with how to actually create large-scale BioScaffolds utilizing the wooden filament. I'm not sure if everyone's familiar with the wooden filament. So it's basically just a PLA filament, but it contains 20% wooden fibers within the filament. And that 20%, although it's a small percentage, is enough for the mycelium to be attracted by and eat through the scaffold itself, which is quite interesting. So currently I'm looking at how to actually create an ideal host system for the mycelium. So I've been testing a number of geometries in order to explore what type of geometry or skin the mycelium is most attracted to. I've been testing a number of shell thicknesses to see what is the ideal thickness for the mycelium to degrade. I've been testing a number of skins. In that sense, porous skin, so testing the geometry of the pores, the density of the pores, testing the materiality of the scaffold. So as I previously mentioned, the mycelium actually is attracted to the wooden filaments of the PLA and therefore degrades the entire scaffold. But I've also been testing a number of additional exotic filaments, such as a different variety of wooden filaments as well. And also looking at internal chambers within the form itself. This has really resulted in a continuous catalogue of forms that evolve in complexity and detail. So this is basically a methodology of research through design. So basically how it works is I'd computationally generate a form, fabricate it, test the mycelium's compatibility with this form, reflect upon the successes and the failures, and then move on to the next design iteration. So as I mentioned, I computationally modeled the form, fabricated it, tested the mycelium's compatibility with it, moved on to the next design iteration. And through this process, I've also been testing different materials, such as PLA, your standard 3D printing material, but also PVA. So PVA is actually a filament that was created as a support material, but what's quite interesting is within this material is that is degrades quite quickly within water. So the mycelium's moisture content degrades this the quickest. And through these experiments, what the main goal is I'm trying to achieve is ultimately to create multi-material scaffolds. So rather than having your one standard material, having a unified form that varies in PVA, wooden filaments, but also PLA in order to really push this concept of this orchestrated process of biodegradability. Here's just some images of the preparation process. It's quite a rigorous process in order to avoid infection or external bacteria seeping through the mycelium. So basically it really begins by fabricating a form, sterilizing the form to remove any bacteria, preparing the mycelium within a variety of syringes or other applications. Once I've prepared the mycelium, I then infuse the scaffolds with the living organism. I then allow it to incubate over, I'd say over a few weeks you start to see results. I'm not sure if everyone's seen the fish tank outside that I hide with the sheep. I do that so in order to incubate the moisture inside that fish tank and not allow any sunlight to come through, because that actually dries up the material. And as you can see here, the before and after the scaffolds, this was obviously day 1, and I believe this was 1 week after implantation. So the results are quite quick. Currently I'm constructing a catalogue of wooden filaments. So I've been testing a number of wooden filaments that actually vary in their fibrous properties. So some wooden filaments that you buy actually only contain 10% wood content, as opposed to some filaments that contain 40% wooden content. The issue is that, you can ask Elliot, because he's laughing. The issue is that the filament containing 10% wooden content prints beautifully because it only contains 10%. The filament containing 40% wooden content is extremely difficult to print. As you can see here, it's about print here, due to its high content of this fibrous property. However, this is what the mycelium is most attracted to. So currently I'm experimenting with what is the ideal filament to print with and what is the ideal filament for the mycelium. And really through this catalogue, my goal is to, as I mentioned before, create this multi-material singular surfaces. So a surface that varies in its wooden content. So maybe an area that would contain 10% and an area that would contain 40%. And just an image here, just showcasing how my vision is to create large-scale prototypes that contain multi-material, multi-skin skins. Here's just a image of the process, so computationally modeling a form, fabricating it, inserting the mycelium, and then really allowing design aesthetics and tectonics to be formed from these experiments. So being an architect, my, I wouldn't say job, but I would say my position within this topic is I have to be very careful not to allow it to become too scientific or too based on materials. So my ultimate goal is to really create a set of design restraints that have resulted from each experiment. So from these variety of experiments and constant form finding techniques, what I found was the mycelium is actually attracted to quite fibrous and delicate forms. So taking inspiration from the sea sponge, the mycelium is attracted to forms that contain internal chambers and this is due to its constant food source for its ability to seep through these micro internal walls. So from these experiments, my idea is to create a range of heterogeneous skins in order to really control and manipulate this process of biodegradability through geometry. And here is an image of the mycelium seeping through these internal walls. And just an image showcasing this controlled process of biodegradability through robotic fabrication and computational design. So the question is how to really apply these scientific experiments to the architectural discipline. So currently I'm working on quite a small scientific scale and also a small scale due to the limitations of the standard 3D printer. But my intention is to really create quite large-scale prototypes in order to prove that these things can actually be applied to the architectural discipline in terms of it could be either an architectural living skin or a structural component for quite large-scale buildings. So my intention is to really create quite large-scale wooden fabricated prototypes. So utilizing, there was a study conducted at RMIT by Francois Rouchet, and he basically utilized wooden pellet material. So pellet material is basically, how do I describe it, Elliot? So like not standard 3D printing on the spool, but
So
yeah.
pellets go into a hopper.
Yeah.
And it's essentially fed through, so they end up being extruded out as a single kind of extrusion line, but it doesn't start on a spool. And
Yeah.
it allows you to do much larger scale things because you don't have to have it pre-spooled. You can just keep filling up the hopper, or have a really big hopper. And,
Yeah, thanks for that.
But
So,
harder to make.
yeah.
Harder to do. You need a more industrial kind of.
So my intention is to now really get out of this 30 by 30 standard set size of the 3D printer and move on to quite large scale prototypes. And in order to do so, my intention is to utilize the UR robots and utilize the pellet material. Another topic that I'm yet to explore within my PhD is real-time robotics. So what I'm quite interested in is this, which I mentioned at the start, this relationship between architect and organism. So throughout this presentation, I've really showcased experiments of how I, as an architect, am controlling and manipulating this organism through computational design and robotic fabrication. But through real-time robotics, I'm quite interested in how the organism may actually affect the overall geometry and robotic movements, and therefore the overall form and aesthetics of the prototype. So currently I'm exploring how this research may contribute to this existing field within architecture. So I'm currently exploring a number of architectural applications. So whether it may be a skin facade, an architectural plugin, a structural element within a building, or are we soon going to create entire buildings made of mycelium that are biodegraded at quite large scale applications. And that's it. Thanks, guys.
Thanks, Nat. Does anyone have any questions?
Yep.
I'm interested in your level of control.
Mm-hmm.
So you create this shape and you stick your mycelium in, but you don't ever really know the way in which they'll move through the shape. Or can you actually quite precisely control that?
Yeah, that's actually a good question because currently I am focusing a lot on form and architectural aesthetics. So as I mentioned previously, you know, I'm currently exploring to create these micro internal chambers to really have a precise control over which direction the mycelium seeps through. But these experiments prove to be quite important to really understand which direction the mycelium will grow and why. And from these experiments, I move on to the next design iteration and say, okay, this is how I want the mycelium to grow and biodegrade. But as you mentioned, the material is actually quite unpredictable. But that's what I find quite fascinating about it. You have a certain element of control over the organism and certain areas where you have to really allow it to grow naturally and randomly.
Do you have a temporal control to go with your spatial control? Can you kind of somehow control like how fast it happens?
Yeah, and that's through geometry, materiality of the scaffolds,
Yeah.
and the type of mycelium that I've inserted with, yeah. Yeah.
I'll keep going.
That's okay.
So someone interrupt. Have you thought about using, and this might be a really dumb question,
Yeah.
but I'm curious anyway. So like edible interfaces are becoming a thing.
Mm-hmm.
And so people are trying to design clever ways of making like uniquely edible shapes that change and all this kind of stuff.
Mm-hmm.
And so as intelligent as we've got so far basically is pasta. Is there a way in
Raw or
which...
cooked?
Well, apparently, if you cook pasta, it changes shape. That was a Clyde paper, I think, 2 years ago from MIT. And I mean, we were blown away.
opportunity.
And so I'm wondering whether there's an opportunity for the mycelium to be edible and your structures to be edible, and you therefore to do something interesting in the edible space.
Yeah, actually, it's a shame I don't have an image of it, but when you implant the mycelium, it actually, because it's still a living form, it actually fruits, and what that means, it's mushrooms actually start to grow on top of it. I think there's an image. Sorry, you see this image here of the mushroom growing on top of the cling wrap bag. That's basically the part that you can eat and pick that off.
Oh, okay.
Yeah, is that what you're asking?
Yeah.
Okay.
And so then is there, so at the moment you're kind of playing, well, you have been playing with wood-doped PLA. Could you instead use, pasta? Could you use something where actually the scaffold itself is also edible? So that like as the mushroom grows out here, I can eat this bit to make it not be able to also then grow out here or whatever? Like, is there some kind of interactive opportunity there?
Yeah, that's actually a good question because it's something I'm quite interested in doing rather than relying on the standard wooden PLA material. You know, current designers have been utilizing, where's that slide? 3D printing with beeswax and 3D printing with a number of, I can't find it. 3D printing with a number of extremely organic materials that you can eat and biodegrade at a more rapid pace. So that's something that I definitely want to start testing within this PhD as well. Yeah.
Speaking of eating
Yeah.
PLA, so the mushroom, the mycelium eats the PLA. That's pretty cool. So
Yeah.
Can we like get rid of plastic by getting mushrooms to eat it?
Yeah, so that's one of the channels that I'm focusing on with this PhD is how to create more sustainable materials through mycelium. And architects have already proven that mycelium can actually replace concrete. So now through this PhD, I'm exploring, how can mycelium actually replace plastics. And they've actually created a number of prototypes. I think IKEA created a mycelium box rather than the standard plastic box they always use. So yeah.
More like your water bottle,
Yeah. I don't know about water bottle, but so yes, this is definitely up and coming research that people have been exploring. Yeah.
So, to chime in on that, my understanding is that PLA specifically is a biodegradable plastic, but there's no way that mycelium is going to grow on PET or ABS or
Yeah.
something like that, like a water bottle. So, because PLA is derived from organic material. It's actually one of the biggest uses at the moment of excess corn and parts from the corn industry in America and China are basically selling husks and stuff that they can't normally use. It goes through this polymerization process where it gets turned into PLA. So it's not going to be the environmental cure for our plastic problems.
Yeah. Not that.
I did see something about this bacteria that will
Yes,
eat
there's been a lot
plastic.
in enzymes and bacteria that will make them kill.
It's like microplastics are a huge problem.
But people are worried about putting these genetically modified bacteria out just into the ocean.
Yeah. I don't have a very big success.
Yeah.
Track record is poor.
So you were talking about sort of going larger scale, right?
Yeah.
So, for example, you were talking about when you had them in the fish tank, you had to sort of cover them up so the moisture would keep, and that would be like sort of ideal growing conditions.
Yeah.
So if you sort of go larger scales, does that also have to translate? Like you'd have to cover it up for a certain time, or how that?
So there's actually two options. I've had many conversations with John about it as well. Thinking about in the near future how to create smart scaffolds. So the scaffold itself actually incubating the mycelium and providing it with its ideal moisture content and temperature, rather than having this external layer of manual glad wrap or something. Thinking about how to create smart forms that actually are the host system for the material.
Right.
Yeah.
Okay.
Yeah. Yet to be explored though.
I guess the other thing is too that I think that you're going to look at is how much the material suits the environment in which it's growing. So
Mm-hmm.
you know, something like this might be more suited to a moist tropical environment or something that has low rain, whereas there could be other kinds of organisms that might work better in dry environments.
Yeah, yeah.
Good question.
Mm-hmm.
Oh, that's fine. So first, I just want to comment on your presentation. It's very visual, and I like the
Thanks.
slides. And I'm impressed by the sheer number of images. That must be a huge amount of work just to find those and put them into these grids. So it's very nice, entertaining to watch. But so my question is about biodegradability. So you mentioned that a couple of times, but I'm not really sure where does that come into the picture? So if you're trying to build structures, where is biodegradable become important, and how are you using that?
In the sense that of creating a form that's biodegradable? Is that?
Well, I mean, you talk about architecture and building structures and things. So my assumption would be that you want it to not biodegrade. So is that right? I'm just not understanding like how where the biodegradable comes into the picture, because you mentioned it a few times. I'm just wondering where does that fit into what you're doing?
So my intention is to biodegrade through the form.
Why do you want your house to biodegrade?
So my intention is to create a form, a set geometry that nature would biodegrade this set geometry and adopt its form. So creating almost like a temporary scaffold as they do in architecture, and allowing nature to adopt this form, and then completely diminishing the scaffold itself.
The scaffold biodegrades, but then
The organism takes over.
Structure is more permanent.
Yeah, exactly.
Okay,
Yeah.
okay,
Yeah.
just wasn't sure about-
It gets more complicated too because the mycelium can die,
yeah.
and like a lot of the materials, it's not right. Like if you buy a chair made of mycelium, it doesn't look right. Or actually, there's one example there where it does, but it can be treated. So there's a company patented the ability to make fake leather jackets out of mycelium, and so it's not like your leather jacket suddenly starts sprouting mushrooms. You go into a dead state basically where it's can be chemically otherwise treated, not dangerous chemicals, but so that it just becomes like regular material like cotton. Like cotton is a material in clothing, for example. Cotton is living, but it's not living anymore, but it still biodegrades as well.
Yep.
But is the material biodegrading, or is it just being eaten?
It's being eaten, so it's basically
Oh, I suppose, yeah.
Yeah.
True. And then my second question is, have you tried it the same thing underwater with coral? Or do you have a sense of like whether it would behave similarly? Because so those coral things, like they grow and then they create really solid, effectively rock, give or take.
Yeah.
So can you create a structure like this, introduce coral, and have the coral kind of do its thing around it? And then introduce mycelium and have it? No, I'm a bird.
No, I haven't looked into it, but yeah, interesting. I will have a look into it.
So it's about so coral grows in that, lots of shipwrecks and stuff.
Yeah.
So, grows around that.
Yeah.
But
What
the
about
coral goes to the
gentrification?
scaffold and itself, which I guess would be the difference.
Yes, you'd have even less control.
Well, the living part of
Coral,
the coral
so-
is just the outer parts, and the rest of the
Yeah.
material is dead.
Could you do something like mimicking what happens in nature with petrification, but compress 100 million years down to a couple of weeks? Something with machines?
With like a pressure chamber.
With a pressure chamber or something?
If it was in paper, you'd say it is beyond the scope.
Just,
And what happens if you mash this up with the silkworm thing? Like, can you introduce it all into one big dome and...
Yeah, Natalie.
Hey, if you guys are cool with me bringing silkworms in, then I'm happy to try it.
Yeah, sure.
Elliot?
I think one of you
Yeah.
But the problem with silkworms in Australia is because they only eat a particular leaf, and the leaf doesn't grow naturally here. So the silkworm industry is actually in Japan. It's a special Mulberry. Yeah. So they can't grow enough material for it to keep the silk production.
It was quite sad actually. My student was doing silkworms as is. So I run Biolab Studio, and each student chooses an organism. One of them chose silkworms, and then for his final, he had just a graveyard of. It was really sad because none of them survived. Yeah, good project though.
Have you thought about putting in a proposal for the new TED building? So the new building they're sticking over there, they're looking for proposals for stuff to put in the entranceway, and like big, impressive looking things. It could be fun to have that kind of be somehow alive and dropping spores on people as they walk in and out.
Yeah, actually the intention is to definitely apply for a range of competitions. That's why currently I think it's really important to begin moving up in scale to prove to people. I think once you start having large scale prototypes, you can actually begin to prove to people that this may be applied to architecture or an installation rather than, the small prototypes that I currently have. Yeah.
Okay. Thanks so much, Natalie. So now
Thanks,
no one has
guys.
an excuse not to know what.
Yeah.
Thank you.
Thanks, guys.